welcome all to the first inaugural Mitochondrial ARS Gene Scientific Symposium. I would first like to thank the organizers, Children's Hospital in Orange County, and then most importantly, Ms. Magee and Ms. Rowland for putting together this foundation, and B, putting together this amazing agenda that we have today for this conference. This really emerged really quickly. I'm really impressed by the speed that this took off. We had an initial conversation with them and Dr. Abdenur and the CHOC team I think just a few months ago, and we got this going together. So I'm really excited. I appreciate the fact that we are able to participate. And I hope that this is going to be a great educational day and bringing us all together as scientists and patients and advocacy groups together. So I'm just going to take a minute to briefly throw in the idea of what these diseases are. Of course, you see speakers with expertise in individual forms of these diseases. But you know, for the patients out there, just to bring it into our mind, what are we actually talking about here? We're talking about a group of genetic diseases that are involved in the translational machinery of mitochondrial proteins. That already sounds quite complicated. So trying to distill it down, each cell in our body has DNA. We have about 35 trillion cells in our body. Each of them, except the red blood cells and platelets, have their own piece of DNA, our genetic code. And that DNA has basically the recipe for all the things that our body does. And in order for that to happen, that DNA needs to be copied, or we call that transcribed, into what we call mRNA, a messenger RNA. And that messenger RNA brings that information from the center of the cell to the rest of the cell. And that information is actually translated into the product, the protein, through the process of translation. And so we are dealing here with a group of diseases that deal with translational machinery. And so what that really means is that in order for this translational machinery to happen, there is a piece of basically enzymes that recognize the individual amino acids that are part of our proteins, and also recognize something called a tRNA, which is a counterpart to these amino acids that recognizes the genetic code on the messenger RNA. So these enzymes are extremely important because what they really do is they guard the genetic code in our cells, and they determine which amino acid binds which tRNA, and consequently how that protein structure actually looks like. If these enzymes don't work, it's fundamental to everything that our cell does and are essentially not compatible with life. And so we have 20 essential amino acids. Consequently, we need 20 different enzymes. Also importantly, this process is happening in two places in our cells. For one, the DNA is transcribed in the center of our cells, in the nucleus, and then the proteins are made through this translational machinery within our cell in the so-called cytoplasm. But then we have these little organelles in our cells called mitochondria that are important for many functions, including energy synthesis, but also doing many other things. And for these mitochondria have their own little piece of DNA. And because of that own little piece of DNA and the small group of genes that they need to have the same machinery again inside them. And so therefore, there is two groups of tRNA synthetases, the ones that are sitting in the cytoplasm and the ones that are sitting in the mitochondria. We are focusing today on diseases that are due to mutations in these tRNA synthetases that are responsible solely for mitochondrial protein translation, for this part. And so the very first disorder that was described with this was described actually before we knew the gene back in 2003. I still remember Dr. Van de Knap from the Netherlands who presented these cases in 2001 at the conference. And she and I were arguing back and forth whether these are mitochondrial disease or not. And this was eventually published in 2003. And then a few years later, she and many investigators together internationally identified that this gene that was involved in this first disease that we call today LBSL. Since then, in 2007, as you can see, we had this first disorder DARS2 described, and in the same year, RARS2 was described. I'm sorry. And since then, we have basically described mutations in every single mitochondrial tRNA synthetase, the last one being WARS2, which was described in 2017. So we now know that mutations in these genes can be associated with disease. Each of these diseases are relatively different and yet similar in some pattern. The important thing is that the vast majority of these mitochondrial telogenesis affect the nervous system, and it's often the central nervous system and in some cases the peripheral nervous system as well. So in the rest of the day, we're going to hear about these individual diseases and the progress that has been done. Important for everybody to keep in mind that this is very recent. In the field of science, things don't happen from bench to bedside quickly. Even though science advances really fast, the process from bench to bedside is often a decade or two and sometimes more. So many of these diseases have just really been very, very recently described, and consequently, there's still so much unknown and so much still to do ahead of us. But I think this is a great way to kickstart this process. I'm going to stop here and hand it over to the next speaker. Thank you.
Hi, everyone. Oh, let me get my video.
Good morning.
Good morning. So I just want to say my name is Ashley Rowland, and I am the co-founder of CureARS.
My name is Desiree Magee, and I am the other co-founder of CureARS. We're so happy to have everyone here, and we've put together an intro video for you to watch.
I believe, yeah.
Hi, my name is Ashley Rowland, and I'm a parent, but I am also a co-founder of a nonprofit called CureARS.
Hello everyone, my name is Desiree Magee, and I am the co-founder of CureARS.
CureARS, the main goal is to raise money for research into a group of ultra-rare genes called Mitochondrial ARS. Our mission is to spread awareness, connect and provide support to affected families, and to fund research for these genes. The purpose of that is to find a suitable treatment option and to ultimately find a cure.
Hi, I am Desiree, Daphne's mom. She is a 6-year-old living with mitochondrial disease. Daphne was diagnosed shortly after she turned 1 year old. Her road to diagnosis, I should say, was rather quick, and it was about 5 to 6 months. She was a normal, healthy baby at birth. At 6 months old, I said, okay, something's wrong. And she said, oh no, it's fine. If she's not sitting up by 8 months, let's go see a neurologist. The blood work showed abnormalities pointing to a mitochondrial dysfunction of some kind. We went to more doctor's appointments than I can possibly remember. They were very lengthy, 2 to 4 hours for each appointment. We were there multiple times a week. So we had actually taken a trip when Daphne was 11 months old. On that trip, we call it, she suffered a neurological regression. There's no real clear definition of that, but in our eyes, what it meant was a regression in the sense that she lost her hands, the use of her hands, arms, and the ability to roll over. So her hands were clenched into a fist. She couldn't push up into, a crawl position. She could not roll over at all. She was very handicapped in mobility, and it was terrifying. But here we are 5 years later and doing the best that we can.
Her disease type is known to cause a progressive brain disease, and it is genetic. And her gene is called AARS2. It has many different names, but it causes a white matter brain disease. And there is no FDA-approved treatment or cure, and it is potentially terminal. When Aubrey was 11 months old, she started having neurological symptoms with every fever. All of a sudden, she had trouble sitting, standing, or walking, and her eyes would start crossing. The first time it happened, we rushed her to the hospital. Her neurologist initially told us Aubrey looks fine. When she was diagnosed, the same neurologist told us there was nothing she could do and to take her home and enjoy our time with her.
Reach up and climb up. Reach. You got it, baby. You got it. You're okay.
On that day of her diagnosis, I was told she could die and end up in a wheelchair, unable to speak, and there was nothing I can do about it. This was not the diagnosis we were expecting. I decided that day that this is not the answer we were going to accept. Her doctors were already giving up on her before she even has any serious symptoms.
Hello, doctor. I need to go home right away. Bye-bye.
The reason we're starting this nonprofit to fund disease research is the lack of funding that's out there, the lack of research that actually exists, the ultra-rare population that we get categorized in, all unfair barriers that prevent, potential scientific exploration of our kiddos' conditions.